Hi, everyone. My name is Derek. I'm a business development manager here at GenoBioTX's. It's a pleasure to welcome everyone to today's webinar. We're excited to have you here with us for today's talk, which is titled Enhancing Your Research, How GenoBioTX's Gene Editing Technology Empowers Research Breakthroughs. Before we get started, I just want to quickly introduce GenoBioTX's. So we are committed to advancing biomedical research by providing innovative and high-quality animal models and services that help accelerate the development of next-generation therapeutics. Our goal is to bridge the gap between basic research and clinical application, and we're really excited to share today some of the insights that can help you achieve that through our products and services. So for today's webinar, we're going to cover the following topics. First, we're going to introduce GenoBioTX's gene editing platform and technologies. We're going to then highlight how these techniques help enhance and advance your research projects. And we'll also cover the history of genome editing and how we utilize modern approaches in our pipeline. At the very end, we'll also have a Q&A session. So if you have any questions throughout the webinar, please feel free to put them in the Q&A chat in the Zoom box as we go along. So to introduce the speaker for today, we have Dr. Yang Yu, who's a business development manager here at GenoBioTX's. Dr. Yu received his Bachelor's of Medicine from Ningxia Medical University in 2014. He then continued and obtained his Master of Medicine in Nutritional Sciences at Harbin Medical University. And then he also obtained his PhD from the University of Florida in the field of nutritional sciences. After his PhD, he started a postdoc in Dr. Jody Babbitt's lab at Harvard, Mass General, where he focused on gene regulation, transporter function, and mineral metabolism. Dr. Yu now acts as one of our business development managers for the Boston and Northeast regions of the US in the Industrial Client Department. And without further ado, let's get started. Dr. Yu, the floor is yours.
Thank you for the introduction, Derek. Hello, everyone. Thank you for taking the time attending today's webinar. And for today's webinar, I'm going to talk about how GenoBioTX's gene editing technology can promote your research projects. And I will use a couple real-world cases showing the different genetic engineered models and how the researcher using these models to perform their study and have high impact publications. So for today's content, first, I will briefly go through the history of genomic engineering, and then I will introduce the most commonly used gene editing techniques. And then I will use 6 real-world cases showing the different genomic engineered models and how the research use that models for their study. And in the end, I will briefly talk about GenoBioTX's service for model generation. Yeah, so let's start with the first chapter, the history of genomic engineering. When we talk about genome and DNA editing, we have to go back to 1953, when James Watson and Francis Crick, they first found out the DNA double helix structure through the extracellular picture, and that opens the door for the genome and for gene editing. And about 20 years later, Paul Berg, which established the DNA recombinant techniques, which is the foundation for genome editing. And just 1 year later, the first genetic modified organism is established by these 2 scientists, Albert Bowie and Stanley Cohen. So what they did is they introduced a frog ribosome RNA into a bacteria and successfully expressed it. That's the first genomic engineered organism. And just 1 year later, the 3 scientists, they established the first genomic engineered mouse by inject retrovirus into the mouse embryo and introduce the virus DNA into the mouse genome. And that's the first transgenic mice. And around 1980s, with the establishment and about mouse embryonic stem cell line and the site-specific homologous recombination techniques, a lot of genomic engineered mouse models, especially for knockout mice, were being generated using the EXL method. And these three scientists, Mario, Martin, and Olive, they received the Nobel Prize in 2007 for their great contribution for the ES-based engineering. And earlier 2000, a new technology, the endonuclease-based technology, such as zinc finger and TALEN, was established for gene editing. And in 2012, the CRISPR-Cas9 techniques was widely used for gene editing. And these two scientists, Jennifer and Emma, they also awarded for Nobel Prize for their contribution of CRISPR-Cas9 techniques. And at GenoBioTX's, we first generate the embryonic stem cell-based gene editing mouse back to 2002. And we received the CRISPR license from Broad Institute in 2018. So far we have generated more than 20,000 genetic engineered mouse models using either these two technology. So that's a brief introduction for the gene editing history. And in the second chapter, I want to briefly talk about the most common used gene editing techniques, the embryonic stem cell method and the CRISPR-Cas9 based gene editing techniques. And also I will compare the advantage and disadvantage between these two knowledge. So for the embryonic stem cell method, first you have to establish the mouse embryonic stem cell line. So the ES cell line, which is derived from the mouse blastocytes, and this cell line require a certain condition for culturing and also a special medium. And then once you have the mouse embryonic stem cell line, and you just need a targeting vector, which have a positive and negative selection cassette, and to introduce the foreign sequence into the embryonic stem cell through the electroporation. And with the positive selection, such as antibiotic selection, we can select the regulate targeted embryonic stem cell line. And once we get the pure population of the targeted ES cells, we can inject the targeted ES cells into blastocytes and then transfer the embryo into a surrogate female mice and deliver the F0 founder mice. So the F0 founder mice, they are chimeric mice, which means they do not have a homogeneous genome. With the edited embryonic stem cells, some of the tissue and cell carries a mutation, which indicates right here. And another part of the tissue and cell from the donor wild type does not carry the mutation. So the next step is to breed the F0 founder mice with wild type mice. If the founder mice also have carry the mutation in the germ cells, then the mutation will be transferred into the F1 founders, and that's to achieve the germline transformation, which can establish the stable modified mouse line. So this is the brief workflow for the ES-based gene editing model generation. And for CRISPR-Cas9 based editing, and it's recently more popular used. And for the Cas9, which is endonuclease, can introduce DNA double-strand break. And so you can think about the Cas9 as a scissor, which can cut in the genome. And so how to achieve the specific modification is through the get RNA, which can get the Cas9 into a specific location in the genome. So the Cas9 is a scissor, and the get RNA get this scissor to introduce the double-strand break in a specific location in the genome. And the editing comes with the DNA repair mechanism. So there are two different DNA repair mechanisms. The first is the non-homologous end joining, the NHEG repairing mechanism. For this repairing method, it does not require a repair template. So right after the double-strand break, there are enzymes to reconnect the break sequence together. However, the repair mechanism is not perfect. It always have some scars, and the scars are small deletions or insertions. We call it indels. And the indels can totally mess up the reading frame, generate the frame shift mutation and premature stop codons. So to achieve that will generate the knockout loss of function of that gene. So usually this non-homologous enjoining will generate the knockout mice. And what if you want to knock in a sequence? Then we can use the second repair mechanism, the homology direct repair, HDR. So for HDR repair mechanism, it requires a repair template. The template can be a double-strand DNA or single-strand oligonucleotides. And with a homologous arm, and we can insert the foreign sequence into the genome where have the double-strand break. So this is the two repairing mechanisms and how it can achieve different gene editing. And for CRISPR-Cas9 based gene editing, it can directly inject the editing material, including the Cas9 enzyme and the GNRA and template into mouse sockets. So here in the left is a picture showing the microinjection working station with the microscope and the microinjector, and the picture on the right showing under the microscope we can have a tube holding individual targets and a micro glass fiber pipette perform the inject with the Cas9 and targeting marker directly into individual targets. After injection, we just transfer the injected targets into surrogate female mice and directly deliver the F0 founder mice. So with these two different strategies, which one should we use for genome engineering and mouse model generation? And CRISPR-Cas9 as a newer technology, it does have some advantages. First is the timeline. It usually requires a shorter timeline for model generation since directly injection of the targeting material into targets and bypass all the cell culturing and screening time. So it's requires shorter timeline and also usually it's cost effective. However, for the cell-based engineering, it's longer timeline and more expensive. And another disadvantage is since so far only the established cell line is on the B6 background. So using the ES-based gene techniques, we can only generate the B6 background mice. And for the CRISPR-Cas9, we can isolate targets from different background and strain, and even we can generate rat models using the CRISPR-Cas9 gene editing techniques. For the ES-based gene editing techniques, there are also some advantages as well, especially when involving large sequence knocking, and the efficiency and success rate is higher when you have a large sequence you want to incorporate into the genome. And for CRISPR gene editing, if this large knocking sequence over 10 kb, the efficiency will be significantly decreased. And another disadvantage for the CRISPR gene editing is the potential off-target effects. So the getter, they can target non-specific targets in the genome. However, what we can do is we can predict this off-target site and perform sequencing to confirm there is no off-target event. So depends on the project, and we can choose either the ESL method or the CRISPR-Cas9 method for the model generation. Okay, so this third chapter, let's move on to the real world cases. So in this chapter, I will have 6 different models using different strategies, generate different genetic engineered models, and how I will show how the researcher using these models to perform their studies. First, let's talk about a knockout mice. So a knockout means the gene is being inactivated. And by doing that, we can replace the gene with the artificial sequence or disrupt the gene sequence directly. And there are 2 types of knockouts. Conventional knockout, which means a global knockout. The gene is globally knocked out in all the cell and tissue. And if you want to only knock out the gene in certain cell and tissue, we can generate the conditional knockout mice and by using the Bray-Flocks recombination system. So first, let's see how to generate a global conventional knockout mice. We can use either the CRISPR gene editing technology or the ES-based technology. For CRISPR gene editing technology, as I mentioned, one guide RNA and Cas9 can generate indels and mess up the reading frame, generate frame shift mutation, or we can use two different guide RNA and Cas9 and to delete a large sequence. And for ES-based targeting technology, we can have a targeting vector with a selection cassette replace certain exons or sequence to achieve the knockout. And for the first case, let's see, for this study, this group of scientists, they studied the transplant arteriosclerosis, which is a major complication in heart transplantation patients. And the model they use for their study is a vascular allograft models. They built this allograft models in different immunodeficient mice, including the FOXN1 knockout mice, which missing the functional T cells. So we generate this FOXN1 knockout mice for them. So first, we evaluate the mouse FOX1 genome sequence, and after evaluation and analyze, we decided to use the CRISPR-Cas9 method and design two guide RNAs and knock out exons 3, 4, 5 of the mouse XOX1 gene. And this knockout also will generate frame shift and premature stop codon to achieve the knockout. So to confirm the genotype of the knockout mice, we perform DNA sequencing. So here is the DNA sequence information. You can see in the knockout mice, there is more than 5,000 BP deletion. And from the amino acid sequence, also showing the mismatch, missense mutation after the deletion and the premature stop codon. And PCR genotyping results also confirm the deletion in the knockout model. And for the phenotypic analysis, they performed a flow cytometry and showing that the T cell in the blood is missing both in the 2 weeks and 4 weeks FOXN1 knockout mice. And in the bottom part is the fluorescent staining of the CD3 marker, which is the T cell receptor marker, is missing in the knockout mice. And what if you want to only knock out the gene in a certain tissue or cell type? So in that case, we can generate a conditional knockout model, which means the gene is only being knocked out in a certain cell and tissue. And to achieve that, we can use the Cray-Loxp recombination system. And this system also can combine with other different inducible systems to achieve inducible knockout as well. And so let's see how this Cray-Loxp system works. Cray, which is recombinase, can recognize the two Loxp sites in the genome and will delete the sequence in between these two Loxp sites and just left one Loxp site. And the Loxp site is a DNA sequence, only have 34 bases. So to achieve this condition knockout, you first need to generate a flux mice, which have the two Loxp site inserted into the target gene, flanking the sequence you want to delete. Then you have a Cray mice. The Cray mice will only expressed in the targeted cell and tissue. For example, if you want to target hepatocytes in the liver, then you can knock in the Cray into albumin gene, and the Cray only expressed with the albumin gene, which is in the hepatocytes, and Cray will recognize the two Loxp sites in hepatocytes and achieve the deletion only in that cell. And combined with the Cray-Loxp system with the estrogen receptor binding site or the tetracycline inducible system, we can achieve the inducible knockout models. So for the conditional knockout gene editing strategy, we first need to generate the flux mice, and either CRISPR or the ES-cell targeting method can be used. And the idea is just to introduce the two Loxp sites, flanking the target sequence you want to delete. So here, for example, if we want to delete exon 2 in this gene, then we insert two Loxp sites, and after recombination, the exon 2 is removed. And using the embryonic stem cell, it's similar. We have the targeting vector carrying 2 LOXP site flanking exon 2, and also have a neomycin for positive selection for targeted ES cells. And after we select the cell, we can remove the selection cassette as well. And with the correct recommendation to remove the target exon 2. So for the second case, which is conditional knockout mice, and this group of scientists, they studied the N6 muscle adenosine, which is the most common mRNA modification and is related to allergic asthma models. They first identified M6A muscle transferase, which encoded by the METTL3 gene is expressed at a low level in the macrophages from the allergic asthma patients. So they want to generate a conditional knockout model by only knocking out the METTL3 gene in myeloid cells. So how to achieve that? We generate the flox mice. This is the mouse METTL3 gene. And after the evaluation, we decided to remove exon 2 and 3. So we use the embryonic stem cell method, have the targeting vector with 2 LOXP site flanking exon 2 and 3, and also have the neomycin selection cassette. And we, after select the positive embryonic stem cell, we remove the neomycin selection cassette. And then we cross this flox mice with LYZ2 cray mice. This LYZ2 cray mice only have the cray expressed in myeloid cells. So after the cray recombination, exon 2 and 3 is removed in the myeloid cells to achieve the conditional knockout of this gene only in the myeloid cells. So here is the genotype validation by PCR genotyping, confirm the genotype of the conditional knockout mice. And also from the Western blot data, also showing that bone marrow-derived macrophages from the knockout mice have significantly decreased expression of the METTL3 protein levels. And then they use these knockout mice to introduce the allergic asthma model by using the cockroach extracts. So here is their experimental plan. And in the allergic asthma models, they found out that the total immune cell numbers is significantly increased in the knockout mice compared to the wild-type mice. And by differential cell counts, they also found out the eosinophils is significantly increased in the knockout mice compared to the control. So what if you want to knock in a gene into the mouse? The knock-in can be a reporter gene, such as fluorescent reporter, or a cray. As we said, if you need the conditional knockout, you need to have the cray mice to knock in the cray into a certain gene. And also, what if you want to introduce a mutation or a human sequence into the mouse? Then we can generate the knock-in mouse for you. So let's first talk about a point mutation. So a point mutation is a genetic mutation where a single nucleotide base is changed, inserted, or deleted from the genome. This single nucleotide change can be synonymous mutation, which doesn't have any effects since it doesn't change the amino acid sequence. Or the point mutation can have severe effects, which change the protein production, composition, and function. If a single nucleotide change lead to a different amino acid, which is the missense mutation, such as the sickle cell disease, a single base pair change generated the missense mutation in the alpha globin and totally influence the protein folding. And also another case, the single nucleotide change into a stop codon and have the nonsense mutation. So to generate a point mutation, we usually use CRISPR-Cas9 method since it's a small size to knock in the mutation. And we usually use oligo donors, carry the point mutation and introduce that mutation into that certain exons. And here is the real case for a point mutation mice. So this group of scientists, they identified a missense mutation in the PLCD1 gene and in the multiple polymetric coma patients. And so they want to study this point mutation in the mouse model, and we generate the mouse model carry the same point mutation. By evaluating the mouse PLCD1 gene, and we identified this point mutation is located in exon 8, and the protein domain information showing that the point mutation is located at the X catalytic domain. So to generate this point mutation mouse model, we use CRISPR-Cas9 method and have oligo donor carry the mutation and introduced into the exon 8 of mouse genome. And to confirm the genotype, the restriction fragment length polymorphism IC showing the mutation and also scavenger sequencing also confirmed that the mutation is successfully introduced in the mouse. And here it's the animal carrying that point mutation. By 8 months of age, this mouse spontaneously developed solid tumor under the skin, and more than 54% of the mice carrying the mutation spontaneously developed the tumor. And a picture on the right showing the scale-like content around the tumor tissue. Okay, so that's a point mutation knock-in. And what if you want to generate a cray knock-in mice to achieve the conditional knockout? And cray will be expressed and under the certain promoter. So there are two different methods to generate the cray knock-in mice. We can either knock in the cray into the N-terminus of the gene or knock in the C-terminus. For N-terminus knock-in, the cray sequence will be knocked into right after the start codon. And by knocking the cray in the N-terminus, the endogenous gene also be disrupted. So we usually use heterozygote mice if the mice doesn't have any phenotype. And if you prefer not to disrupt the endogenous gene, then we can knock in the cray in the C-terminus. And usually we have another cassette such as self-cleavage peptide, 2A peptide, and errors to achieve the parallel expression of endogenous gene and the cray. So for these cases, the scientists, they want to study the G protein coupled estrogen receptor GPER positive neurons in the retroventricular region in the brain. So the model they generate is an optogenetic activation model to only activate the GPER positive neurons in the brain. And also they want to deplete this GPER positive neurons. So to achieve that, we generate a GPER cray mice. And the knocking strategy is straightforward. We use CRISPR-Cas9 method to knock in the cray sequence into the C-terminus of the mouse GPER gene. And first, to achieve the optogenetic activation, what they did is they inject the cray mice with a virus, AAV virus, which conditional express the CHR2 and mCherry only in the GPR positive cray mice. So the CHR2 is a light-gated ion channel, and it will be switched on with the implanted optical fiber. And the ion channel, when it's stimulated by the optic fiber, it releases ion to stimulate the neurons. And also the mCherry will be expressed to locate the neurons under the fluorescent image. Here, this is the image indicating the expression of the mCherry only in the GPER positive neurons. And after the stimulation, they find out that the mouse is significantly increased the sensitivity to thermal, hot plate, tail flick, and mechanical stimulation. And then next step, they want to ablate the GPER positive neurons. So to do that, another different virus is being injected into the GPER cray mice. And this virus, A virus, it will express mCherry and also conditionally express the diphtheria toxin. So the toxin only will expressed in the GPER positive neurons. Here, this is the data. After inject the virus into wild-type mice, the toxin won't be expressed, only the mCherry is expressed, indicating the neurons. And after inject the virus with the cray mice, the cray will have achieved the conditional expression of the toxin. So you can see the mCherry signal is diminished in the cray mice. And another most common model is overexpression model. And for the overexpression, the traditional way is just to randomly insert the gene into the mouse genome to achieve overexpression. But recently, a more accurate way to do it is to specifically knock in that sequence into safe harbor locus. And by means safe harbor, which means the knocking in that location won't affect the endogenous gene. And there are some common used safe harbor locus, including the ROSA26, H11, and COL1A1, and they are located in different exons. And for a global overexpression, the strategy is straightforward. Just knock in the promoter with the coding sequence of the gene of interest and validation sequence into the safe harbor locus to achieve the global overexpression. If you want to only overexpress the gene in certain cell and tissue type, then we can combine with the Cray-LoxP system. So the knocking will be the promoter with the LICL cassette, two LoxP site flanking a stop cassette, and followed by the cDNA sequence of the overexpressed protein and validation signal. And so before Cray recombination, the overexpressed protein is stopped by the stop cassette. And after Cray-mediated recombination, the Cray-positive cells have the recombinase to remove the stop cassette and only have that target gene expressed in that certain cell and tissue type. So for this case, I want to talk about a really interesting model. They use two different recombinase systems, both the Cray and Duray recombinase system, to mediate the cell lineage tracing and the cell subsite ablation in vivo. So here is their strategy to generate this model. They knock in the sequence into the safe harbor ROSA26 safe harbor locus. They use the two recombinase system. So the CAG promoter is a strong synthetic promoter. And the first is the Cray-LoxP system, two LoxP sites flanking the stop cassette. And then the second is the Duray-ROX system. So the two ROX sequence is flanking the second stop cassette, and then it's the TD tomato and the diphtheria toxin receptor and the WPRE cassette. This cassette is just to stabilize the mRNA to achieve higher expression level. So the Duray-ROX system is similar as the Cray-LoxP system. So the Cray only recognize the two LoxP sites and remove the stop cassette, and the Duray only recognize the two ROX sites and remove the second stop cassette. So if you only have a Cray expressed, remove the first stop, and the TD tomato DTR won't be expressed since the second stop is still here. So similar, if you only use Duray to remove the second stop, the TD tomato DTR also won't be expressed. Only when both Cray and Duray expressed, the stop cassette is removed, TD tomato and DTR will be expressed. So this cell will be labeled with the TD tomato, and once you administrate it with the toxin bind to the receptor, leading to the cell dies. So in this study, their strategy is to targeting cardiomyocytes, and they use these two, the ACTB Cray mice and TNI3 Duray mice. These two genes are specifically expressed in cardiomyocytes. So they cross this cray and doré mice with the Rota26 knock-in mice. And the ACTB cray will remove the first stop site, and TNI3 doré will remove the second stop site. And the TD tomato and DTR will be expressed specifically in the cardiomyocytes. And when the mice reach 6 weeks old, they will inject with the diphtheria toxin or PBS and analyze 2 days after the administration. Here in panel D is showing the mouse movement tracking. So here you can see after the DT administration, 2 days later, the mouse moving is diminished. And they also confirm the heart injury after the toxin administration. Since the cherry also expressed, so they are able to use fluorescent image to see the fluorescent image of the whole mount heart or a section of the heart tissue. And here you can see the TD tomato is expressed inside the cell and the diphtheria toxin receptor expressed on the cell membrane. And with the toxin administration, the cell leading to the cell dies and the fluorescent signal is diminished. Okay, so the last case is a humanized mouse model. So recently, a lot of humanized models is being established to replace a certain mouse gene with a human gene. And the reason to do it is since the protein will be human protein, and this model can be used to analyze any human-specific test articles. For example, if you have a anti-human antibody, then you can use the humanized mouse to evaluate the antibody targeting the human protein. And what I want to talk about today is humanized TRF1 mice, which is a popular model recently for neuroscience studies. TRF1 physiologically function is to mediate cellular iron uptake. So iron is binding to the chaperone transferrin in the circulation, and the transferrin receptor is a cell membrane protein which can bind to the transferrin, carry the 2 irons, and through endocytosis go inside the cell. After iron release, the transferrin and transferrin receptor is recycled back to the cell surface. This is called the transferrin cycle. So the physiological function is just to bring the iron inside the cell. However, since the transferrin receptor 1 is also highly expressed in endothelium cells of the blood-brain barrier, so scientists can use the transferrin receptor as a target to deliver their therapeutics across the blood-brain barrier into the brain. So we generate this humanized transferrin receptor 1 mice by knocking the human transferrin receptor 1 gene into the coding exon of the mouse gene, which is at exon 2, using the embryonic stem cell method. And we have this neomycin selection cassette to select the positive targeted EXL and the tyrosine carriers for negative selection. And then we remove the neomycin selection cassette. And for the humanized mouse model, mouse transferrin receptor 1 gene is knockout, and the human TRF1 protein is expressed. So we did the validation by Western blot, showing the human TRF1 is expressed in homozygote mice in the brain, liver, and the small intestine. And also fluorescent staining also indicate the human TRF1 expressed in the brain and intestine tissue. We then performed an in vivo study to evaluate the TRF1 binding antibodies. We gave the TRF1 binding antibodies into our humanized mice and also compare with the control antibody. So the TRF1 binding antibody is enriched in the brain and decreased in the serum, which indicate this TRF1 antibody is bound to the human TRF1 in the blood-brain barrier and being successfully transported across the barrier into the brain. And so that's just showing that it can be used as a test article drug delivery system to do a central neural system delivery. Okay, so that's all the 6 different genetic engineered models and generate at GenoBioTX. So in the last chapter, I want to briefly introduce our gene editing services and how we can help you with your model generation. So at GenoBioTX's, we can do customized model generation. We can generate any genetic engineered mouse model, including all the strategy models I've been showing before. And we can also do rat model generation and the cell line engineering as well. And we have so far established more than 20,000 already established models. These models are our catalog mouse, including some point mutation mice, humanized models, such as human immune checkpoint immune checkpoint humanized models, and some immune cytokine humanized models for therapeutic anti-human therapeutic evaluation. And we also have immunodeficient models and immunosystem reconstituted models, and also cray mice, reporter mice, different fluorescent reporter mice, and global knockout and conditional knockout mice. These mice are already established and they are ready for your research. Besides for model generation, we also have a strong pharmacological team. They can perform in vivo, in vitro pharmacological, pharmacokinetic, and dynamic studies to do drug evaluation, efficacy studies, toxicity, and we can perform these studies in-house using our animal models. And other supporting services, including breeding services, some embryo freezing recovery, and we also have zebrafish and C. elegans platform as well. And as I mentioned, we have generated more than 23,000 models, and with 10,000 models directly ready for research. And our cell line have reached out more than 500 genetic engineered cell lines. And these cell lines, including tumor cell line for generating syngeneic tumor models. And for our R&D capacity, we can perform more than 4,000 R&D projects per year. And what is the workflow for customized gene engineered mouse model generation? So we start with the background evaluation and project evaluation and design. So we will understand what you want to perform for your study and what kind of model will be the best suit for your study. And we will do the evaluation and bring up with gene editing design. And once you confirm the design, then we can sign the contract and start the project. And once we achieve each milestone of the project, we give you updates for each step, so you will know the model generation processes. And once we confirm the genotype of the model, we deliver the model to you. And we can also provide after-sale services, including the genotyping protocol if you have any problem with genotyping, and also breeding plans, breeding schemes. We can help you out with that as well. So the whole model generation services is to make sure the model you generate can be used for your research projects. And so if you are interested in any new models for your research, you can just go to our website, GenoBioTX's.com, and search our category for your model. Most likely, we already have the model for you, and you can directly purchase that models. In other cases, we can customize and generate that model for you. And so that's how we can help you with your research projects. Yeah, that's all for today's webinar. And if you have any questions, I'm happy to take in the questions. Thank you.